Obesity is an epidemic in the United States and a leading cause of multiple major health problems. Therefore, an effective treatment is needed. Obesity is caused by increased food intake, which is controlled by various circuits and signals in the central nervous system. One such signal is ghrelin, which is released from the stomach. Circulating ghrelin levels are elevated before a meal and increase food intake. Ghrelin's intake stimulatory effects are attributable to its action on its receptor, growth hormone secretagogue receptor 1A, otherwise known as GHSR1A, which is located in the brain. Ghrelin increases food intake when injected into the hindbrain ventricle and into specific hindbrain nuclei, including the NTS, but the endogenous role of GHSR1A in the hindbrain is not well understood. This project tested the hypothesis that hindbrain GHSR1A plays a psychological role in the hindbrain to increase food intake, and tested this hypothesis by chronically knocking down GHSR1A in the NTS and acutely injecting the endogenous GHSR1A antagonist LEAP2 into the fourth ventricle. In the first experiment, we used a bilateral NTS injection to knock down the GHSR1A and implant an injection cannula. After the procedure, we measured baseline 24-hour chow intake and body weight and determined that GHSR1A knockdown did not impact the baseline chow intake or body weight. We measured baseline chow intake in light and dark cycles and observed that GHSR1A did not impact light versus dark cycle chow intake as well. We then injected 0 or 150 picomoles of ghrelin into the fourth ventricle and measured chow intake. We observed that GHSR1A knockdown attenuated ghrelin-stimulated chow intake. From previous experiments in the lab with CCK, we knew that hindbrain ghrelin blocks CCK-induced intake inhibition. We then further hypothesized that the knockdown would increase CCK sensitivity. To test this, we injected 0 or 0.5 micrograms of the intake inhibitory GI satiation signal CCK subconsciously and measured short-term chow intake. We found that GHSR1A knockdown did not impact CCK-induced intake inhibition. In the second experiment with a second group of rats, we implanted an injection cannula into the fourth ventricle and verified the placement by 5 Tg and 100% increase in blood glucose. We then injected 0, 150, or 1,500 picomoles of LEAP2 and measured chow intake and body weight. We observed that LEAP2 decreased chow intake and body weight. Results show that while the chronic NTS knockdown of the GHSR1A did not result in baseline differences in chow intake or body weight and only mildly attenuated the intake stimulatory effects of ghrelin, acute hindbrain injections of LEAP2 significantly reduced food intake and body weight. Together, results suggest a role for hindbrain GHSR1A in the endogenous control of food intake.